Good afternoon, and thank you for joining the RLS Foundation's webinar titled Iron and RLS. My name is Carla Diezenkowski, and I'm the Executive Director of the RLS Foundation. The surge in the use of web-based programs during COVID-19 health crisis has created a strain on the system because everyone's doing what we're doing. So if you're experiencing technical difficulty, switch to audio. Use the telephone number listed on the invite, as it uses less bandwidth and you'll be able to listen in that way. We thank you for attending and appreciate your patience and understanding during today's webinar presentation. Before we begin, please be aware that all attendees will remain on mute during this hour. We thank you for the questions submitted with your registration. Today's presenter, Dr. Connor, will answer as many questions as time allows following the presentation. Dr. Dermer will also join us for that part two to help fill some questions because we had quite a few submitted, and I thank everyone for those. After the webinar, you will receive an email with a link to the webinar recording. The webinar recording will also be made available in the members only section of our website. If you're not a Foundation member and if you would like to view this and other recorded webinars, we invite you to become a member today by going to rls.org. As always, individuals suspecting that they may have RLS should consult a qualified healthcare provider. Information offered in this webinar is for informational purposes and should not be considered as a substitute for the advice of a healthcare provider. Dr. Connor is a distinguished professor of neurology, neural and behavioral sciences and pediatrics, vice chair of neurosurgery research, and director for the Center of Aging and Neurodegenerative Diseases at the Neuroscience Institute at Penn State University on the Hershey campus. Dr. Connor is a leading researcher in the field of iron and RLS. Dr. Connor, we thank you for sharing your time and expertise with us today. I now invite you to begin the presentation.
Thanks so much, Carla, and thank you to the audience for joining in from around the country today. And I look forward to sharing some updates on our research program, hopefully some insights into the relationship between iron and restless legs, and then particularly look forward to your questions at the end. I know this is a little bit of a different type of approach. I always encourage people to ask questions as we go along, but I know we can't do that in this environment. So but please do consider your questions, and we'll be able to address as much as we can, as Carla said. So first, I need to start with a disclosure. So I have a company that's been developed around the treatment for iron deficiency using ferritin, and I will be mentioning ferritin in this talk, but it's a little bit different. We're not advocating anything in terms of our product, but I'm the chairman of the board and founder of this company, which is known as Citro Biosciences. So a little bit on the background, the pathophysiology underlying RLS may be related to decreased iron in the brain in individuals with restless legs compared to normal. And we began these studies these ideas by autopsy studies, and maybe I could take a moment now just to tell to acknowledge the folks that particularly in the early days of really getting the RLS Foundation started and enabling us to study the brains from postmortem, from autopsy, and a lot of what we're going to be able to talk about today, and a lot of what we understand in general came from this incredible donation, this gift that these early individuals gave us. And so at every opportunity, we always want to express our profound gratitude for those individuals that enabled us to share the information we have with you today by looking actually at the brain after they passed away. So in the early autopsy studies, there was a protein profile in the neuromelanin cells, and these are the cells that are the ones that make dopamine in the brain. And so these are the ones that we particularly focus on. We'll look at some MRIs where you can see that part of the brain. in the living subjects, but we were able again to look specifically at and directly at the neuromelanin cells in our RLS patients and subjects, and that's really when we began to develop this idea that the brain itself in the RLS population was iron deficient or had less than normal amounts of iron. And one of the key findings that we had, so there's this protein called transferrin receptor. So transferrin is the protein that transports iron throughout the blood, throughout the body. So that's its name, trans and ferrin. So transporting iron or carrying iron. It has a receptor that's on almost every cell in the body, and then including the cells that make up the blood vessels in the brain. And that's one of the ways, and we'll look at this in a little more detail in subsequent slides, but that's one of the ways that the transferrin itself brings iron into the brain. And normally that, if you're iron deficient, that will go up. That's one of our really good indicators when a cell is iron deficient. The receptor for the protein that's going to bring it iron goes up, and that makes sense. But in the RLS, everything we were looking at was telling us that there's not enough iron in these neuromelanin cells, these dopamine-making cells in the brain in RLS. This receptor did not go up, and that, again, was one of the first signs of not only supporting the idea of iron deficiency, but this might be an example of a place where we should be looking to understand how these cells became iron deficient. Why did they not increase their receptor expression? And the other part of the interesting finding there was that the tyrosine hydroxylase, so that's the enzyme that makes dopamine. And so dopamine, just to back up a second, dopamine is the primary neurotransmitter that we're interested in restless leg. So the neurotransmitter is a sort of a scientific way, medical way, if you will, of talking about this chemical that's released by one nerve cell to allow it to communicate with the other cell. So it transmits the neuronal response. So neurotransmitter. So tyrosine hydroxylase is the most important enzyme in making dopamine, and we focused in on that because it absolutely requires iron for it to work. And what we found is that it was actually elevated in the restless legs patients. So again, if there was an iron deficiency, we thought it should have been decreased, but it was actually not only not decreased, but increased a little bit. But that also went along with some of the animal models that we studied of iron deficiency. So, sort of right away, we were learning something new in terms of some of the basic neurobiology of these cells, if you will. But it also gave us an indication that the dopaminergic system could be intact. And that wasn't our problem. So unlike Parkinson's disease, where we have a decrease in dopamine, that wasn't what we were running into in the RLS population. So just sort of for orientation purposes, and I think you can see my arrow as we move it around here. This was one of the early images that we had from an RLS patient. And actually, I've been showing this slide for a long time, and it made me think, and I looked down here. So this study was published in 2001. So this was really one of the first studies to kick off the idea of iron deficiency or insufficiency in the RLS brain. So let's start over here, normal. You see this area that's lit up here. This is called the substantia nigra. So this is where those neuromelanin cells, those dopaminergic cells that we're interested in, this is where they reside. So and you can see that it's nice and bright there. It's nice and bright because there's plenty of iron. So iron would appear very bright in this picture. This is another neighboring part of the brain called the red nucleus. It's also iron rich, so we can see it nicely there. So this is what normal should look like. So if we go over here to an RLS patient, this is that same area of the brain right here. You can see this little line, and it's not bright at all. And this is that neighboring red nucleus that we talk about, this round area right here, also not bright at all. So there's clearly a significant lack of iron in the RLS brain compared to normals. And just as to go over here to the right side and look, this is an autopsy picture that was stained for iron, just so we could see. So all the dark blue is iron. So here is this substantia nigra with its nice dark blue staining, and this would be correlate to this area over here in the brain. So the substantia nigra, this is what it looks like with a lot of iron in it on the neuroimage, and this is what it looks like at the time of autopsy. And again, the circular red nucleus is also there, so you can see it over here on the image. So this was, as I said, part of what kicked us off to say, well, we need to go in now and look at the autopsy studies to begin to directly examine the brain to understand how, we should be like this. How did we end up like this? And so I mentioned dopamine, and that's what the DA stands for up here. And I mentioned, so there's no loss of these tyrosine hydroxylase neurons, no loss in the substantia nigra, or another area of the brain that we and others have looked into. Bill Ondo in Texas has looked into. So there's no great loss or significant loss of these kinds of neurons. And that's the reason that's important to mention, is that so restless legs is not a neurodegenerative disease. People with RLS ask frequently, is this Parkinson's? You're giving me a dopamine drug and I have, these restless sensations in my leg, and is that sort of a precursor of Parkinson's? The answer is pretty much no. So again, Parkinson's is a neurodegenerative disease. When we look at a Parkinson's brains, these tyrosine hydroxylase neurons, 80% of them are usually lost, at least in the Parkinson's brains, and even more. So there's no loss in RLS. So the system is intact. So we just have to learn how we can modify the system, tweak the system to make the restless legs go away. The dopamine neurotransmitter system, as I mentioned before, is basically intact. So tyrosine hydroxylase was elevated. Dopamine, the neurotransmitter that we're really interested in, also seems to be elevated. So again, in Parkinson's, you're trying to replace the lost dopamine with the treatment strategies. That's not a problem in RLS. One of the things we did see is a decrease in receptors. The dopamine itself has a number of different receptors, D1, D2, and so that just means one of the kinds of dopamine receptors. D2 is lost or is decreased in the RLS subjects, which is again consistent with some of the D2 agonists that are used to treat RLS. And it's also consistent with what we see in our animal models of iron deficiency. So again, these animal models are informing us and helping us to better interpret what we see in the RLS brain as really indicative of iron deficiency or iron insufficiency. So this is an actual picture from one of our autopsy studies, and this is, again, helped us in supporting the idea of iron deficiency in RLS. So these are those neuromelanin cells, or the tyrosine hydroxylase cells. They're one and the same that we've been talking about in the previous slide. So this is what it looks like in an autopsy specimen from an RLS brain. This is what they look like in a control brain. And the blue here is ferritin, this protein here. And ferritin is around when a cell has to store iron. So iron can't sort of hang out by itself. It can cause problems if it's not sequestered in a protein. So ferritin is sort of this little cage. It literally looks like a cage or a ball, and the iron goes down inside of it so that the iron, when it's loose, doesn't cause any problems. It doesn't cause any problems when it's stuck down inside the ferritin protein. And again, then the protein gets broken down when the cell needs iron, and iron can become available. But this tells us, when you see all of this blue in these normal cells, here's an outline of a nice normal cell here, and all of this blue in the cell, we don't see any blue in those cells in the RLS brain. So these guys in the RLS brain are not storing iron. And so again, that's certainly strongly suggestive that these are iron deficient. So how did we get there? How and why is the RLS status in the brain low? So we had to go kind of back to the beginning and ask questions like, well, what's the mechanism for getting iron across the blood-brain barrier? And when I say the BBB, or the blood-brain barrier, what's different about the vasculature, the blood vessels in the brain from any other organ, is that the cells that make up the blood vessels are joined by what we call tight junctions. So anywhere else in the body, the blood can kind of freely get in between the cells that make up the blood vessels and get access to the liver, to the kidney, to the heart. But in the brain, that doesn't happen. The only way blood can get access to the brain is to pass through the endothelial cell, is the name of the cell that makes up the blood-brain barrier. So that means that iron and any other nutrient doesn't get free access to the brain. It has to pass through the endothelial cell. And if you're familiar with cancers, that's one of our big challenges in cancer. That's why all these drugs that we have that are available to treat systemic cancers don't work in the brain, because the way these endothelial cells, the way these blood vessels are configured in the brain, it keeps everything out. And unfortunately, in that case, it's keeping these drugs out so that they can't get access to the brain tumor, or if a cancer is metastasized to the brain, the drug can't get access to the cancer cells that are in the brain. So this blood-brain barrier is very important, because it means that, if you happen to eat something that disagreed with you the night before, and your liver can take it up and help you detoxify it, it doesn't get it into your brain and cause you significant problems. But at the same time, as we said, the trade-off there is that you have to have transport systems in place to get nutrients and things that you need into your brain. So how does iron get into the brain, and how is that movement of iron regulated? And if it is regulated, what are the signals? And the other reason I spend a little bit of time on this slide is that, Carla and the Restless Legs Foundation have spent a lot of time talking to their congressmen, trying to increase awareness of restless leg syndrome. And one of the things that we like to point out with this study is that we were able to get funding from the NIH for these very basic fundamental questions. How does iron get into the brain? Very fundamental question, but very critically important question in terms of all aspects of neurobiology, in terms of the developing brain in childhood needs an adequate supply of iron. There's too much iron in the brain in Parkinson's disease and in Alzheimer's disease. So really, what came out of a basic question to understand iron transport in restless legs has led to a couple of very nice NIH grants for us. And it's one of those things that, as you talk to your congressman, you approach them certainly from the standpoint of, we need to learn more about RLS, but this is one of the areas, this isn't going to be just relevant to RLS. We're going to learn a heck of a lot. And you'll see, again, as we kind of go through the slides, a lot of the things that we've learned because we asked a very fundamental question became relevant to us in studying RLS. So the beginning question, the really basic fundamental question that we had to, in science, we like to call it a paradigm shift. So everybody thought they understood how iron got into the brain. They said, well, here, here's the blood on this side, and this is my schematic of an endothelial cell. So this would be one cell making up the blood vessel of the brain. They said, well, transferrin bound to its receptor on this side, and then it dumped off the iron on the other side. It's not hard. And so we looked at that model, and then we made a couple of important discoveries. We said, well, here's ferritin. Ferritin's in the endothelial cell. That would mean that the endothelial cell is storing iron, as we talked about before. And the other key part of this is that endothelial cells are very, very busy. They have a lot of stuff they have to transport. So they have a lot of these mitochondria, the little energy producers of the endothelial cells. And there's a lot of them. And you know what? One of the most important elements that help the mitochondria use their oxygen and generate energy is iron. So if this system works, then these guys don't work. And so there's got to be a way of getting iron into the endothelial cell. And again, just a fundamentally important cell biology question that came out of these questions that we started asking about restless legs. And this is the conduit model that people had embraced prior to our starting this work, which was that simply iron went from the blood into the brain, carried by transferrin. And finally, we were able to convince the scientific and medical community that that this can't be true given the presence of the mitochondria, especially in the cell. So I'm sorry, I'm going to switch orientation on you just a little bit, but it's the same idea. So here's our endothelial cell, the yellow box here, and now we have the blood on this side, and we have the brain over here on this side. And I'm sorry, but I have a panel that's kind of blocking my view. So on the far right of my slides, I'm afraid if I shut it, I'll turn everybody off. So so pardon me if I'm not pointing to something on the far right of the slide. But basically, this was what we were just talking about, that everybody believed, that transferrin bound to its transferrin receptor, and it came out the other side. One of the other important discoveries we made after we started looking into this transport idea was the presence of this protein here. It's called DMT1, or divalent metal transporter. And the reason it's called divalent is that it likes to transport iron in its divalent form. And so once we found that this protein was expressed in the endothelial cells, in the blood vessels that make up the brain, then there was no question that the iron came off of transferrin when it got into this little vesicle, and that this protein did its job and allowed iron to come out of the vesicle and put it into the endothelial cell, where it could go into ferritin, it could make the mitochondria work, and this vesicle then recycled back outside of the cell. But then our big question became, okay, well, we got iron into the cell, now we still have to get it out. So we did a good job arguing that this happens, but now how do we get iron into the brain? Because obviously we know it gets there, and if we do get it there, how is it going to be regulated? So in order to study the transport, we had to go to a cell culture model. So this is growing cells in a dish and studying as best we can the mechanisms that we can then take back to study in our animal models and in the patients. So this is like a big picture. This is a kind of a well in a dish, and inside this well we put this insert that you can see here. And that way we can grow the endothelial cells across the bottom of the insert. And they form the tight junctions, so they look just like a blood vessel. And then up here in the apical part of our chamber, we can put whatever we're interested in studying to see if it gets transported. So for example, we would put iron up here. And then down here in the basal part of the chamber, we would put whatever we're interested in that might be signaling to support the iron transport. So the first study we did, we got cerebral spinal fluid from monkeys that were either iron deficient or they were iron sufficient. And we put this down in this basal chamber. And the cerebral spinal fluid, I think everybody might be familiar with, but this is the fluid that bathes the brain and the spinal cord. It's a fluid that if you have a spinal tap or a lumbar puncture, that's the fluid that people are, that your healthcare provider is removing. So the CSF is down here, and we put iron in there. And again, the only way for the iron to get from the top part of the chamber to the bottom part of the chamber is to pass through, not between, but pass through these endothelial cells. And so this got us really excited that there was some type of regulation going on. So if you could see here, so this is, we have radioactive iron in the chamber, and we counted it as it came through. And here's what it should look like normally. This is what the iron sufficient cerebral spinal fluid from the monkey, from an iron sufficient monkey. This is what the profile looks like there. But if the monkey was iron deficient, then look, there was a lot more iron that came through. So there was something going on in that CSF that was telling these endothelial cells to let more iron in. So this was really our first example of a transport, of a regulated transport for iron across endothelial cells and into the brain. So then we went on to another study where we really wanted to ask questions about the IV iron compounds that are being used. And so we modified our model just a little bit. You can still see it's a nice well here, and here's the chamber. But now these endothelial cells that we were using that make up, again, the blood vessels of the brain, these are now human cells. Before we were using cells from bovine for the most part. But we worked in collaboration with a really bright young scientist named Ethan Lipman at Vanderbilt, and Ethan helped us to what he was able to do, working with another colleague, Aaron Bowman, who's now the chairman of biology at Purdue. But between the two of them, they were able to take skin fibroblasts and sort of retrofit, if you will, the skin fibroblasts to look like and behave like the endothelial cells that make up the blood-brain barrier in the human brain. So really exciting technology, and again, work that came out of the ideas that we wanted to understand what was going on in the restless leg. So again, up here in the apical chamber, the top part of the chamber, we put our compounds of interest, and then we measured what came through down here. And again, you can see more nicely the endothelial cells are in green, the blue is the nucleus of the cell, and you can see that there's no space in between these cells. The only way, again, something can get from here to here is to pass through the cell, just like in the brain. And so this is what we found, which again started to look to be very interesting. So these are all the different IV iron or representative of the different IV iron compounds. And so what we did was put each of these into the top chamber, and we measured how much iron came into the bottom chamber. And what you can see is for all of the IV iron compounds, here's no treatment, if you can make this out across the bottom. So for all of these different, for all of these different compounds, there was basically no iron that got from one side of the blood-brain barrier model to the other. We had transferrin in there as the, that's our biological standard. We knew that was transported, and indeed it was. And we had also ferritin because of our interest in looking at this as a new idea for iron delivery. We saw that was really very robustly transported. But in the context of these IV iron compounds that people are getting and responding to, but they aren't themselves getting into the brain. And just to kind of put that in a percent total, I know this is very busy and bouncing all around, but we just wanted to see that see folks to see that we're really only looking at, less than 0.2% after 24 hours. So after putting these iron compounds in that apical chamber and waiting for an entire 24 hours, or an entire day, there was still less than 0.02% of these of the iron that got through. So basically, they're not getting through. So these compounds don't get into the brain, but they work. We're not saying they don't work. And so this was a study done by Erica Unger, who was at Penn State, and now she's on the faculty at Lebanon Valley College in Pennsylvania. And she did a study with our colleagues that are familiar to many of you, Richard Allen and Chris Early at Johns Hopkins. And they looked at an iron sufficient, and this was an animal model, an iron sufficient, and this was the control. This was an iron deficient animal, and this was an iron deficient animal that got one of the IV iron compounds. In this case, it was multiside. And what you can see is that after both 3 days and 10 days of post-treatment, if the animal was getting this IV iron compound, that the iron content went back to normal. So this is where it should be. This is what happens when it's iron deficient, and this is what happens 10 days after the treatment. So the iron went back to normal, and that's great. And this ventral midbrain is the part of the brain in the mouse that's very similar to the substantia nigra, that area of the brain that we were first talking about at the beginning. So really good news, these compounds work. So even though I was showing you that they don't themselves get into the brain, they do increase the brain iron content. And that's the important take-home message. We'll look into some mechanisms in the next couple of slides. But equally important, if you look at these other regions of the brain that didn't become iron deficient or significantly iron deficient, they also don't take up a lot of the iron compound, even when it's after 3 and 10 days post-treatment. So this is really important. Somebody had sent in a question about, do you worry if you're on an IV iron treatment about iron getting to areas of the brain that you don't want it to get into? And areas like the hippocampus, the things that we worry about with too much iron sometimes in Alzheimer's disease. So the data here, again, this is an animal model, but the data here are pretty strongly suggesting that the iron is going to go into the areas of the brain that it needs, but not into the area of the brain that don't need additional iron. And again, that's obviously screaming at us regulation, which is, again, one of the points, as I said in the previous slides. What we were able to say is, there is regulation and there's local regulation, and this supports that argument. So we have a big brain and lots of different places in the brain, and but the iron is getting to the places that it's needed. So if the IV iron compound's not getting into the brain, but it's working, what might be happening? And sort of the best working idea that people have right now is that the iron gets taken up by cells called macrophages. And macrophages literally mean big, as in macro, and phages, as in eat. So they're really called the big eaters. And their job is to eat everything, to phagocytose everything that you kind of don't want. So they can even phagocytose bacteria. If a cell starts to die in any part of the body, they can take up, they can help remove that cell so they can again they do their job, which is to eat up whatever they don't want, whatever the body doesn't want. So the idea, maybe the working idea, is that sort of this first pass of these different iron compounds are taken up by these cells, and it's not a surprise. That's kind of their job. So these cells will take up the iron, but then what happens? And that was a question FDA has had, that we've had scientifically. It's like, okay, if you just filled these cells with iron, are they going to explode? Are they going to work fine? Are they just going to sit there and have so much iron in them that they can't work very well? So we decided that we would grow these cells in dishes again, and then treat them with a different IV iron compound. And I know this is kind of a data heavy slide, but I think it's kind of easy to understand. So we put the macrophages in a dish, and so we grew a bunch of them. They're sitting in a dish, and then we just added these different iron compounds into the dish and waited 24 or well for different hours. You can see across the bottom here. And then we took what we call the media. We took the solution out of the dish that the cells were sitting in, and we just figured out how much iron is there, because that meant the rest of the iron must have gone into the cell. So there's two things that are really interesting in this slide. One is, here's our the ferric citrate, I think that is. No, I'm sorry, the carboxy maltase. But you can see all of the cells that have an A over top, they all took in about the same amount. And so that, it wasn't like, there was a bunch of iron going in. They all took in sort of, again, a regulated amount. And these two compounds took in less. And so, again, and they were right about the same. So, very interesting that all of these behave in a fairly similar manner. And so we looked again at ferritin inside the cell. Remember, ferritin is sort of our indirect measure of iron storage. And this was, again, very telling. So these two compounds here that left more iron in the media also had less ferritin. So there was less iron being stored with these two compounds compared to the other compounds, who again were all pretty similar and all pretty similarly took up the same amount of iron. So this was a good part of the story. Number one, that the macrophages could take up the iron. They didn't take up more iron than they could handle, and they seemed to be putting it in the right place biologically, which meant storing it in ferritin. So now we've got iron and ferritin in a macrophage. So again, that's all good. It's being put in a place where it's not, the iron's being put in a cell that can handle it, and it's being put in a protein inside that cell that can also handle it. So we feel very good about the whole biology under the intravenous iron compound. You're not releasing iron, massive amounts of iron into the body that's going to cause damage. We talk about oxidative stress and reactive oxygen species, and iron is under normal biological conditions can be somewhat toxic in pushing the oxidative stress and could in fact kill some cells. But so this was again made us feel very good about the compounds that we were looking at, and that they were again being handled correctly, not only by the right cell type, but handled the right way by the cell type. But now what do we do? So we've got the iron in the cell and in a nice protective protein. Then what happens? Well, it looks like one of the things that happens is that the iron can come back out, and it comes back out again in our protein ferritin. So this was somewhat of a new finding, that ferritin was actually being released by cells. And this was, again, like I said, a somewhat new discovery. And to remind you that what we saw crossing in our blood-brain barrier model before, the ferritin was a preferred route of transport of iron, at least in the blood-brain barrier model. So now to go back to the slide, these compounds that put iron into the cell are now coming out as ferritin. And ferritin is, as I said, the preferred compound for delivering iron. So one of the questions we want to ask going forward as we evaluate the efficacy of the different iron compounds is how much of them are being converted to ferritin, and is this the way that the iron is actually getting into the brain? Because again, as we showed before, it's not direct access by the compounds themselves. So another big question has been, can we predict the iron uptake into the brain, and who's going to respond to treatment? And the current standard for deciding if people should be treated with an iron supplement, an IV iron, is the serum ferritin level. And you know, many of you that are on iron supplements and getting treated with IV iron know that if your serum ferritins drop below 100, then you are in discussion with your primary care physician or your RLS doc about whether or not you should get an iron supplement. One of the reasons is that serum ferritin is thought of as representative of what's going on in the body in terms of its iron status, but it's not always the best indicator of iron status. Its levels bounce around a lot. If it's low, then you feel pretty confident that iron status in the body is low, but it ranges a lot. The other protein we were interested in was hemoglobin. Hemoglobin is that protein inside your red blood cells. It requires iron, and its job is to deliver oxygen to all cells, including the brain. And I think this is going to be a really important aspect going forward. We did one of these webinars talking about limited oxygen capacity of the brain in RLS, and we continue to try and look at that. But remember that the brain only weighs 2%. It only accounts for 2% of your body weight, but it uses 20% of the oxygen. So when we talk about taking a walk for your cardiovascular health, you're also doing this for your brain. Your brain is going to take up as much oxygen as and it's intolerant of lack of oxygen. So getting oxygen to the brain is a big deal. And so we kind of want to pay attention to the team and what's going on in restless legs. So let's go back to our model here. And this again is our apical chamber. And these are again are the cells of that are now endothelial cells. And I'm going to show you a slide I think is, we just got this paper accepted, and it was just part of a new NIH grant that we got that generated a lot of interest. And I think this is really going to impact the way we think about iron utilization and think about treatments in the RLS population. So the important point I need to make here is, remember, these endothelial cells came from Ethan Lippman in Vanderbilt. They had no idea about restless leg syndrome. They have no idea about the CSF. The CSF that we got for these studies came from David Rye and Lynn Marie Trotti at Emory University, and from Richard Allen and Chris Early at Hopkins. So the endothelial cells, they don't know anybody they didn't know the people down here, and obviously they don't know what's going on up here. So these endothelial cells are totally naive to any of the measurements we were going to make. And so what we did was we put the CSF from our RLS patients and control patients down here, and we put the iron up here. And then we looked to see what happened. Well, what happened was that there wasn't a lot of difference in the amount of iron that was transported between the RLS and the controls. And importantly, there was no correlation with serum ferritin. So it didn't matter where your serum ferritin levels were. It had no influence on the amount of iron that was going to be transported across the in our endothelial cell model. So, and again, remember that right now, serum ferritin levels are one of the driving indicators of whether or not to treat with IV iron or not. And anecdotally, and some data are out there to tell us that there's no predictive value. So you can't take no predictive value associated with serum ferritin. So you can't go and say, well, this person had serum ferritins of 20, so they responded immediately, or they responded within 5 days of getting IV iron treatment, while the person that had serum ferritins of 100, they didn't really respond much, or they didn't really, it took them 2 weeks to see a response. So this really supports what's going on in the clinical picture, is that serum ferritin doesn't help us in terms of understanding or predicting the transport of iron in the blood-brain barrier model. What surprised us is that hemoglobin does. And so what this says is that the higher your hemoglobin levels, the more iron that you're able to transport into the brain. So what this is telling us, we think, is that if your hemoglobin levels are low, and these are not anemic levels, these are just low and normal, they're telling us that the first thing that the body says is, I have to have enough hemoglobin to transport enough oxygen to all the organs that need it. So that iron is making sure that there's adequate hemoglobin for oxygen transport, and that includes the brain. So the brain is sending, and remember, this was the CSF from these patients. The CSF somehow knew what the hemoglobin level was. The endothelial cells didn't know. They came from Vanderbilt, but the CSF that came from the same patient that had the hemoglobin measures somehow knew what that hemoglobin measure was, and it told the endothelial cells, no, the iron's got to go somewhere else until I get, until I start getting my hemoglobin levels up. So this is, again, telling us that the brain and all organs, but especially the brain, is saying, I'm okay being a little iron deficient, but I'm not okay being a little oxygen deficient. So, as an iron guy, that kind of breaks my heart, but it makes sense in that again, we know the dependency of the brain on oxygen. So as we try to treat the RLS population and anybody with the IV iron supplements, we really need now, we're learning that we really need to pay attention to those hemoglobin levels to help us hopefully identify better who's going to respond to the RLS or to the IV iron treatments. So we can summarize saying that brain iron uptake is regulated at the level of the blood-brain barrier. The blood-brain barrier is not simply a passive conduit. And again, that information came about because we were asking some basic questions about what was going on in restless legs, and as a result, we're changing the way the entire scientific and medical community think about iron getting into the brain. And your work with congressmen and your politicians, local and federal, to support restless leg syndrome. This is one of your big arguments, is to say, again, what I'm trying to get you to do is going to help RLS patients directly, but it's going to help us understand a whole lot of new things. The IV iron compounds that I mentioned don't cross the model of the blood-brain barrier, but it looks like they're handled very effectively by these cells called macrophages, and then processed into the typical way for iron to cross into the blood-brain, cross the blood-brain barrier and get into the brain. And as we said, the important new information here is that hemoglobin and not serum ferritin may be the best indicator of how well the iron is going to get into the brain. So with that, I'm happy to take questions and show you a picture of my currently very empty medical center.
Wow, amazing. I didn't realize it was so large. But amazing. But I wanted to thank you so much. What a great presentation, and also for talking about our advocacy work. Currently, we have 5 members of our Scientific and Medical Advisory Board funded by NIH research. That's Dr. Connor, Dr. Trotty, Dr. Koo, Dr. Richards, and Dr. Early. And this all happens because our constituents pick up the phone, visit their congressperson, or call um, or actually go to Hill Day. But this year we're going to actually do an RLS Advocacy Day virtually. So, join us on April 16th for this webinar, and we're going to give you the how-tos. And this is a really important part because this is how we get legislation through that, recognizes the needs of the RLS community. And it also affects policy within the agencies as well. So, we spent a lot of time on the Hill, as you say. Dr. Winkleman and Dr. Dermer spent extensive time on the Hill with staff last year, talking to, our policymakers. So do make time to join us for that April 16th webinar. So I do have Dr. Dermer joining us as well to help field some questions. So I'll go ahead and get started, Dr. Connor. So after an iron infusion, what criteria is used to recommend a subsequent infusion?
That's a good question. Maybe Jeff wants to weigh in on that. I mean, one of the things that are certainly monitored are the serum ferritin levels to see how the response has gone as an indication of, again, how the systemic levels are of iron are changing. Jeff, do you have more specific recommendations?
Absolutely. Thanks. And again, Dr. Connor, this was wonderful. It helped me to conceptualize some of the changes that I see in clinical practice. So very, very, very fun for me too. Yeah, the majority of people that go through any sort of iron therapy, whether it be oral or IV at this point, are followed in a pretty systematic manner. We look at ferritin levels as an indicator of potential benefit, just given what we've known related to the lower iron status or ferritin status in patients with RLS. But we also look at things like iron saturation, and total iron saturation in the blood is a helpful indicator of sometimes iron overload, which is something we also have to keep a close eye on whenever we're doing iron therapies over time. And there are a few folks out there with hemochromatosis-based genetics that increase the likelihood or chances of that happening. And so at around a 2 to 3 month period after starting oral iron therapy, or within the first couple of weeks after an IV iron therapy session, we like to check ferritin levels as well as the iron saturation. And then in some cases, we also look at CBC just to see what hemoglobin levels are doing as well. Those things do take a little bit more time given the different compartments of iron in the body, but certainly we look at a number of other variables depending on the individual's risk factors as well. There are some people that come into iron therapy who may have kidney issues or diabetes, and we have to be more careful with IV iron and iron therapies in those cases, simply because they can have more of an effect on kidneys in particular, and increase some risk factors for diabetic patients.
And then to piggyback on that, there's a question. It says, are there any medications that may interfere with iron uptake?
Interfere, well, actually, it's not so much medications that interfere, but it's food products. If you're looking to increase uptake in the gut in particular, we know that iron has difficulty crossing the blood-gut barrier, the gut-to-blood barrier, as Dr. Connor mentioned. So anything that actually would interfere with that, there are certainly drugs that are interfered by iron over time, things like antibiotics, quinolones, and tetracyclines come to mind, but also some of the bisphosphate interactions that occur as well with things like Fosamax. They can lower the levels of those antibiotics or bisphosphates in some cases. It's also been associated with lowering some of the HIV integrase inhibitor activity as well. So it's not something we worry about in terms of a regular issue, but we certainly take that into consideration when folks have other medical conditions or medicines they're taking.
Okay. All right, Dr. Connor, does having RLS affect the way that the body absorbs iron, not just the brain?
Yeah, I mean, that's a really good question. And one of the things we've been learning so much is that we actually don't understand and appreciate the iron absorption at the gut to the extent that we might think that we do. And everything that Jeff was saying about what limits iron uptake from the diet is certainly true, but trying to understand the movement of iron across the, it's the same type of barrier to some extent, trying to get things from the gut into the blood. And so the answer is we don't know. There's nothing, and we don't know because we have limited knowledge in the context of how well the iron's being regulated. We know some aspects of the regulation, but how that is impacted by restless legs, we don't at this point.
Okay, next question is for you, Dr. Dermer, and it's how and when should iron tablets be taken and at what dose, and what is the optimum ferritin level?
Well, the last question first, we're really not sure what an optimum ferritin level is. As Dr. Connor indicated, we, for many years, had an impression that perhaps ferritin was a helpful guide with relationship to who would benefit from iron. But again, that's now in question. Certainly hemoglobin plays a role here. So it's really hard to say what an optimum ferritin level really is when you're talking about RLS, but we do know that the associations from epidemiologic studies in particular have shown that people with ferritin levels lower than 50 initially is where we were implementing therapies. That's risen to 75, and now we're really contemplating under 100. So as time goes on, we're learning that it's really not a specific number related to the ferritin level. It's really a helpful guide, perhaps, as related to the therapy we're providing. And so that brings us to the type of therapy. And if you're using oral iron supplements, and I would reference a recent publication I think everyone might be interested in, that was, I think, yes, Dr. Connor was on this publication as well. It's in Sleep Medicine from 2018. First author was Richard Allen, evidence-based consensus practice guidelines for iron treatment of RLS. And in there, they do talk quite a bit about the different oral versus IV strategies. And the idea that, you'd probably want to start with oral iron therapy if your total saturation of iron is less than 45. Again, that's one of those things we test for. To start with an oral therapy like ferrous sulfate, which is usually a 325 milligram tablet with vitamin C twice a day. We're taking a total of that once a day, is sort of the consensus recommendation at this point. But there are a number of other ferrous and ferric compounds that may be more well tolerated for some, such as gluconate containing compounds, as well as fumarate and a number of others. And those all provide a certain amount of elemental iron per dose, but using vitamin C, ascorbic acid, changing pH, which I think Dr. Connor can tell us more about what exactly happens. I believe that does increase the divalent ionic components of ferrous state, so it's actually able to be transported by the DMT1 receptor. But that is something that we know to be a helpful component of oral iron therapy. And then avoiding food and other things that might actually scavenge iron from the gut and not allow it to be absorbed. So we really recommend not using dairy, not eating meals that have a lot of collagen in them, perhaps, that also may sequester iron in the gut as well during your therapy.
And if you'd like access to that article, it's open access. You can email us at info@rls.org and we'll get you the link. Jim, next question for you. How can you have iron cross the blood-brain barrier and not have it build up in your circulatory system?
The so there, there's a normal amount of iron that's always circulating, and then Jeff mentioned the looking at the transferrin saturations, the iron saturation. So the body, for the most part, is very good at regulating how much iron is circulating at any given time. So it will keep the iron in liver and in other organs where it's not bothersome, while it really strives to maintain a healthy level, if you will, of iron transporting. So you can build up your iron stores without worrying, and have adequate amounts to get iron to transport across the blood-brain barrier into the brain without worrying about having to sort of overload your blood or your serum levels with iron in order to get adequate brain iron supply.
Okay, next question is, I had almost eliminated my symptoms with diet change, but now I'm pregnant and my RLS is worse. Why would that be?
Yeah, so that lovely child you're carrying is also one of the greatest parasites that exists. So the child is growing rapidly and developing, and that's where this important regulation of iron is such a high level requirement. And so the baby is fighting you for every atom of iron that you have, and will mostly win. So that's probably why the RLS is back, and FDA suggestions or regulations for iron uptake increase about almost 3 times for pregnant women compared to a non-pregnant. So it's just the taxing that the developing fetus is putting on the system is probably just not being able to be met by the current diet that you're using.
Okay, Jeff, did you want to add in any or?
No, I mean, pregnancy is it's a very difficult time to manage RLS in cases where you're managing it even with drugs and medications that you can't take during pregnancy. So iron becomes one of those therapies that is exclusively important for pregnant women. And as Jim mentioned, this is a group that's at much higher need. But then again, there's also a balance with pregnancy itself, because we know that if we have high levels of hemoglobin in women during pregnancy, that is also sometimes associated with outcomes that are not so good for the baby. So there's a balance between supplementing to treat RLS symptoms, but also not driving up potential hemoglobin levels, which it generally does not happen, but it's something that we theoretically have to keep an eye on throughout the pregnancy.
And we have 2 diet-related questions coming up. So the first one is a vegetable form of iron a reasonable and safe way to increase iron levels? And then is it better to get your iron from fruit or supplements? So I'll let you both take a stab at those questions.
Well, I'll just say that and because Jeff already mentioned, one of the things to be careful about with most of the vegetable types of iron is that there's usually a lot of phytates around, some collagen around, so that type of iron is actually challenging to absorb. And so if you're getting enough and there's no associated symptoms, then that's okay. But if once your body has a challenge, then vegetable or fruit-related iron availability is so limited that it's going to be hard to get adequate amounts from those 2 sources alone.
Yeah, I always think of it, unfortunately, as a little kid who loved Popeye. That spinach was probably not the thing providing him with the iron. It was probably the iron can he was eating it from. But yeah, I concur with Dr. Connor that most of the recommendations we provide are with supplemental iron and vitamin C, not really with dietary iron, other than potentially heme that comes from things like liver and meat sources that may be one of your only dietary sources for absorbable iron.
Okay, next.
Carla, just on a personal note, but to amplify what Jeff just said, I grew up also loving Popeye, and I've always wanted to write a commentary that asking the question, was Popeye a fraud? But because of my love for Popeye, I've never been able to do it. So.
Yeah.
I'm with you on that, Jim. I will not participate in taking down Popeye.
All right. What are the dangers of iron transfusion in the elderly, and how long does an iron infusion typically last?
Yeah, iron infusions in general is a pretty similar issue across different populations, which is you can have a slight immune response to it, an autoimmune response to the iron itself, which it's really an inflammatory response that can be controlled with antihistamines and pretreatment as well with anti-inflammatories, which we typically do in most cases when we start IV iron. The other thing we do is we use IV iron in small doses as a test dose just to see what the effect is in an individual, since things are idiosyncratic when it comes to these reactions oftentimes, but they're very few and far between, and they're well controlled, as the rate of IV iron itself can be tuned up or down depending on the individual. We tend to use more fluids with populations that may be more vulnerable. We even do this with children with IV iron therapies, and it really is a very effective mechanism for kids. The older populations, because of potential kidney issues, we may slow the rates down as well as use more fluids to maintain a good flushing of the vascular system during the procedure itself.
Okay, and then another question about diet is, what is the relationship of gluten intolerance / allergy in regard to low ferritin and iron? What can be done about this?
So I mean, there's quite a few inflammatory conditions in the gut, like colitis and Crohn's and other inflammatory issues related to the gut that iron is also an inflammatory. It's basically it is part of your inflammatory response. So having higher ferritin levels when you're sick is a normal event, and having inflammation as a chronic problem can actually increase that as well. So it's hard to say that giving iron as a therapy for RLS in someone with other gut issues will make those other gut issues worse, especially given the smaller doses we're using. But it's possible that in that population you'd need to follow them more closely for side effects.
Okay. Last question. I have anemia, but ferritin levels in the high hundreds. Is either likely to be causing my RLS?
So that having an IV, so I'm sorry, I kind of missed the beginning of that. It just went in and out on me.
Oh, I'm sorry. I have anemia, but ferritin levels in the high hundreds. Is either likely to be causing my RLS?
Okay. Well, anemia itself, meaning that you have either a hemoglobin. So is it macrocytic, microcytic anemia? First of all, that's one of the things we have to talk about. So what's the cause of your anemia? Not all anemias are the same. Microcytic anemia associated with iron deficiency would be a type of anemia that we would see typically with patients who are complaining of RLS. Macrocytic anemia, not so much. As far as I've seen, it's reported, of course. But that's a different deficiency issue, usually with B vitamins. So I would have to know a little bit more about that individual to give you a good response, but generally anemia that's microcytic in nature is iron dependent, that may be related.
Okay, and then can we sneak in one more question? As far as oral supplementation, is liquid form versus pill form better?
Whatever tastes better to you, it typically
Okay.
works. The benefit of liquid, especially with kids, is they'll take it, and also it's often combined with vitamin C, ascorbic acid already, so you don't have to add additional minerals or vitamins.
Okay. Well, thank you so much for joining us today for this webinar, and Dr. Connor and Dr. Dermer. I know we're all sheltering in place, and it's nice to get together with our community and have a little session to learn a little more about iron and RLS, because that's just such a hot topic for our community. But I hope everyone's safe and well, and we'll talk to you on the 16th for the next webinar on advocacy.
Thank you, everyone. Thank you, Carla. Thank
Thank you.
you.
Great.
Thank you,
Thank you.
Jim.